So hello everyone, I want to make this video to show you a bit of a teaser about my coming PhD defense. To see if any of you are interested, you can message me on Twitter or the email below, and I will send you the direct link to attend on November 30, 2020 at 2 p.m. Paris time. The main topics of my PhD are going to be cell migration, actin, and blebs. And the specific topic is the stabilization and motility mechanisms of blebs in confined cancer cells, which I've done under the supervision of Mathieu Piel in Institut Curie in Paris. As maybe some of you know, there are two main mechanisms that allow membrane protrusion in migrating cells, one being the keratocide, at which the actin directly pushes against the membrane by polymerization, so that and this is done because the actin filaments are directed in a way and are polarized so that the membrane is protruding only at the front. And another one, which is very different, is on blebin cells that you can see here on the right. And blebin cells, rather than by polymerization, they advance the front membrane by the intercellular pressure. So you have a buildup of a contractile cortex on the rear of the cell that will contract the cell and will push the front like if you were pushing one balloon, for example. I'm studying the migration through blebs, which is rather the second type. Blebs have been described as a link to apoptosis traditionally, but we know more and more that they are important for cell migration. And typically the blebs have been described in the literature, and this being a topic, rather hot topic for biophysics, for example, they last in the number of minutes. But in the past few years, we have discovered that when cells are confined below 5 micron, or around that for HeLa cells, for example, they can make blebs that instead of having a typical lifetime of minutes, they rather last hours, and they don't retract. They maintain a constant flow of cortex from the front of the cell to the rear of the cell, and this cortex, this super fast cortex, thanks to some friction, is able to propel the cell forward. And eventually led them to migration, like you can see here on the right. So the topic of my PhD is what is the difference between these blebs that retract within a minute and the ones that last for hours, and what is the mechanism for them to stabilize and migrate eventually. And here just I will give you just a hint on some results that might be interesting, and some cool movies. If you know more, you can come for my defense. For example, we saw that in migrating blebs, if we track at each point of the contour the actin density and the edge velocity, you can see that you have two transitions. At very low actin densities, the cortex is protruding, but very quickly as you increase the density, you go to a state of frozen cortex, meaning you don't protrude or retract. And then at very high actin densities, which correspond to the back of the cell or the back of the bleb, is when you start to have contraction. And the threshold from a protruding to a non-protruding cortex is rather sharp, so you have a very fast transition. Now, we don't know a lot about how actin really works in cells on the molecular level, but these three states are really reminiscent of what's been described for in vitro system, where we can access better the molecular dynamics of actin. For example, I need to cite some excellent work from the lab of Kinneret Keren from 2015, or a paper from this year, where they have seen that in extract of Xenopus, they can reconstitute cortical flows with turnover, and they see that the length scale of the flow depends, for example, on the turnover and the cross-linking of actin. Or some other work from the lab of Gijsje Konderink in Netherlands, where they make like little gels of actin at different cross-linkings, and they see that the length scale of the contraction or the perturbations that the motors do on the cortex, it depends also on the rate of cross-linking. So from in vitro models, this has been rather well studied, and people have proposed that there are these 4 states at different network connectivity and motor density. One which is an active solution, where actin is not able to transmit any force because it's not connected enough. And then as you increase density or cross-linking, you start to have a pre-stress gel, meaning that the gel are transmitting the stress over long range, but they are not contracting yet. And as you increase contraction, you can go to 2 different states. One of global contraction, where the entire network retracts or tears apart, or local contraction, where you have local um, centers of contraction and turnover. And this being based on percolation theory, which is a range of mathematical theories and work that describe how you can define the global behavior, so the microscopic behavior of a network, like rigidity or conductivity, based on the molecular properties when you change the concentration of links. And very importantly, this has been described in vitro system, but has never been applied to a motile system or a living cell. And this is mainly because we were not able to see actin filaments at a very molecular level, because normally the cortex is just so dense that unless you use electron microscopy, so you kill the cell because you need to freeze it, you don't really see that at this detail. But thanks to the combination of our marker and a very good microscopy setup and microfluidics, and also our system, because you have a range of cortical density, we could achieve an unprecedented resolution to see how actin assembles from a bare membrane to a very dense cortex. And what we realize is that, as you can see here, that in front of stable protrusions, what we just saw in the cell before a low resolution, the protruding side or the front correspond to gas-like state of the cortex, meaning that the filaments are like wiggling around and diffusing, and they are not dense enough to transmit any forces. And that in fact, anytime the filaments go until the membrane at the front, as you see here, it causes retraction. So just a few filaments at the front are sufficient to perturb the membrane and cause the retraction. And also, for example, at very low densities, if there are not enough connection, actin filaments, they can stay in the front and they don't flow until they are connected to the network. And you can see here in the chymograph, the filaments don't move until they are connected. So if you trace, if you measure, for example, the average front flow or the flow in the back, you can see that in the front you have this on-off behavior, so you have this pulsatile flow, but in the back, because it's always connected, you have a constant speed. What we think is that these force percolation theories, they can explain very well the dynamics of the cortex that we see in the front of stable blebs. You can see, for example, here I color in blue and I skeletonize the parts of the actin network that are connected to the base. So meaning to the part of the bleb that is contracting and where myosin 2 concentrates. And you can see that only when the clusters become blue, so that they connect to the main network, is when they are going downwards. We could reproduce some of these dynamics by simulations that take into account the constraints of percolation. So when filaments are, for example, sparse enough, and you have a gradient of nucleators, under these conditions you can reproduce the waves that we see in blebs. And this is more or less the steady state picture that we are proposing for stable bleb, where in the front you have a loose cortex where filaments are not able to transmit any force, so you don't see any flow, they are just diffusing. And then rapidly, as you increase the density, so you go through a critical percolation density, which depends on the geometrical properties. Then you go to a cross-linking cortex, where the filaments behave like a solid. They're able to transmit forces, but they are not contracting yet because there is no myosin II there. And then only in the base of the bleb is where you have both high myosin II and actin. And this contracting cortex here, you start to see a convergent flow, and it's where network disassembly happening. What we also proposing, and I hope to tell you more during my defense, is that we think that these four states can provide us with a model for bleb shape or cell shape during this stable bleb migration, such as in the front of the bleb, where you have this round hemispherical cap, corresponds to the state of an active solution, so that the membrane is just round because it doesn't suffer or doesn't see any force on the cortex because the actin density is too low. Then the sleeve part of the bleb that you can see here is correspond to the state of a pre-stress gel before the onset of contraction, and then the back of the bleb corresponds to the onset of contraction or global contraction here. The fourth state of local contraction, we normally don't see them in blebs, but we can promote it, for example, by inhibiting up to 3 or 4 means that decrease the density of the cortex. So I hope you like this short talk, and if you like it and you want to talk about it more, you're more than welcome to contact me. And I can also give you a link to attend my PhD defense on Monday, November 30 at 2 p.m. Paris time. It will be online. Hope to see you soon.